Alright, so on this one we're going to talk a little bit more about the accessory organs. All these organs we're going to see are going to kind of put their materials into the digestive tract. None of them have the bolus or the chyme technically traveling through them, but these are going to be the organs that, again, to me, put some more of the materials into the mixing bowl that ends up being the duodenum. So a lot of these are part of what we call the biliary apparatus, which is kind of the liver and the bile network that is part of that. So we're going to see the liver actually makes bile, and it's going to have two main ducts that drain bile from the liver. You have left and right hepatic ducts that merge together to form a single common hepatic duct that we're going to see kind of comes down to a point and merges with another duct that goes up to the gallbladder called the cystic duct. Beneath that, we have the common bile duct that goes down and empties into the duodenum. Goes through a little opening on the wall called the hepatopancreatic ampulla, and there is a sphincter there that's going to regulate the flow of these materials in or out. And it makes a little bump on the wall where both, like I said, the bile and that pancreatic juice are going to enter. There is a secondary duct for the pancreas called the accessory pancreatic duct that has a smaller bump called the minor duodenal papilla that lets small amounts of pancreatic juice enter the duodenum separately. So this kind of shows you a lot of these parts of this biliary apparatus. Again, you can see the left and right hepatic ducts merging to form the common bile duct. Merging to find the common hepatic duct, which merges with the cystic duct right here, which if we're not releasing bile into the digestive tract, the bile will actually travel into the gallbladder where it is stored and concentrated. If the gallbladder contracts, it's going to push stuff out the cystic duct into this common bile duct, which again will empty into the duodenum, and this is just down from the stomach. One of the things we need to talk about, and this is one of the few spots we can actually talk about this, is liver is involved in a lot of different things. We honestly don't go into nearly as much depth as we could on the liver. It is highly involved in metabolism of a number of different proteins, lipids, other things like that. Really plays a role in almost every macromolecule in terms of its storage or conversion of a lot of these. It's also, when you look at any of the cats or anything else here, it is the largest internal organ of the body. You can see it has 4 separate lobes to it that are separated by these little ligaments. I'm not so worried that you know which lobe is which, but you can kind of see here is the liver. It is that upper right quadrant just underneath the diaphragm. I mean, kind of a deep brownish red if you're looking at it within an organism. You can see 2 main lobes to it, the left and right lobes. Right lobe is much larger than the left. If we look at it on the underside, you do have a couple smaller lobes that are part of this quadrate lobe and the caudate lobe, which are just these little parts here and here. So it is separated into a lot of different lobules, and really what the liver ends up doing is it is a filtering organ. It, similar to the kidneys and the spleen, is a filtering organ. It is going to be composed of a bunch of these little separate lobules that have a large capillary network within them, and really all the blood that's coming back from the digestive tract is going to have a first pass through the liver. The liver cells are referred to as hepatocytes. These are generally simple cuboidal epithelium, and we're going to see you get these what are called portal triads that have some of the different tubes of some of this here. So we're going to see we have bile ducts, we have what is called branches of the hepatic portal vein, and you're going to have a main artery that's traveling up this stuff as well. Within these capillaries are very open capillaries, so they're that sinusoidal type here. This allows a lot of macrophages and other stuff to mix in here. Again, because this blood is coming from the digestive tract, if we have ingested something, this is an area where we could have pathogens getting to the body rather easily. So it makes sense that the liver is screening a lot of this blood as it comes back from the digestive tract. If you see right here, this is a sinusoid. You can see it's a hexagonal structure lined with these epithelial cells along here. We can see you have these portal triads of a bile duct, an artery, and a vein that are going to release blood into here. This blood moves to a central vein. All these central veins come back together sooner or later to be a hepatic vein. and this is what's going to actually go into the inferior vena cava. Like I said, all that blood is moving from the outside in. Why what we have going on here is a lot of these cells are making what is called bile, and that's actually going to be moving in the opposite direction out towards that portal triad in the bile duct. And again, all this blood is moving through these hepatocytes, and the bile ducts are kind of sandwiched in between this. So you can see the repeating units of the liver here with these hepatic sinusoids, and again the portal triads at some of the corners of these kind of hexagonal-like structures. Again, all these central veins from all these sinusoids are going to merge together at some point, and that's what becomes a hepatic vein, which is going to be what is leaving the liver. Along the way, a lot of these cells, as they modify stuff that is in the bloodstream, they are going to generate bile. Bile is going to move in the opposite direction out towards that bile duct, and those bile ducts are all going to merge together to become the left and right hepatic ducts, which are going to carry that bile down to the gallbladder. Again, all this blood from the digestive tract is going to go through the liver first. So this is another one of those portal systems, in this case the hepatic portal system. Everything that is absorbed from your digestive tract, the blood is going to merge into one main vein called the hepatic portal vein, which is going to go into the liver and pass through another capillary bed before it gets back out and gets into the inferior vena cava and is going to go back to the heart. And part of the reason for this is the liver is doing a lot of detoxifying, and as well as just overall a lot of metabolism of these different things. Like I said, if we were doing a metabolic chapter, which we don't tend to do in A&P, it's involved in carbohydrate metabolism. So this gluconeogenesis, it's also involved in forming glycogen and storing glycogen. A lot of lipids are modified by the liver and stored within the liver. We store iron within the liver. Proteins can get modified and changed by the liver. We also do a lot of protein synthesis in terms of clotting proteins and plasma proteins. We store fats, we store vitamins, we store glycogen here. Again, on top of that, a lot of immune cells and other stuff looking at this. The liver is one of the main organs that is going to remove a lot of toxins and drugs from the system. Alcohol being an example of one of those. In terms of digestion, the only thing it's really doing is synthesizing bile, and that bile, part of that is getting rid of those hemoglobin byproducts and sending that down to the digestive tract. The other thing that this can do is can actually activate vitamin D formed by the skin. The gallbladder, the bile that is made by the liver, it is going to get sent down here. It has a number of layers to it, a very thick muscle layer though. It has some folds within it that can definitely allow this gallbladder to distend in when it's filled with bile. It's connected to the biliary apparatus by that cystic duct, and there is a sphincter that's going to control the flow out of here. And again, that bile goes from those left and right hepatic ducts down to the common hepatic duct, joins the cystic duct, takes the bile up to the gallbladder. Downstream of that is the common bile duct, which is going to take any of the bile from the liver and the gallbladder down to the duodenum. Normally, the bile, when we're not doing digestion at that point, it's going to be stored in the gallbladder. Can be concentrated a little bit there, but this contains water mainly, some bile salts, and a number of other things that we're going to see in bile. And you can see kind of the idea right here. When it comes time to actually release this, we have chyme that's released from the stomach. And one of the things that happens when we're having this release here, we get a distension of that duodenum, and it causes those intestinal endocrine cells, those enteroendocrine cells of the intestines, to release cholecystokinin, also known as CCK. CCK, we already know in terms of the stomach, tones down its activity. But CCK is going to act on the gallbladder, and it leads to contraction of that gallbladder, which is going to cause the bile that is stored within the gallbladder to be basically expelled out and down into the duodenum. And that opening in the duodenum relaxes and lets that enter out into there. What is this actually going to do? Well, it helps the digestive system because it is what is called an emulsifier. It reduces surface tension between fats. So when we were just doing a little Zoom talk, fats, if you've ever had Italian dressing or anything else like that, you know the fats try to stay all together and will separate out. Because our digestive enzymes are mainly in water, things that are high in lipid are going to try to form one big fat glob. What bile does is it takes, instead of having one big fat droplet, it's going to make that one big fat droplet be a bunch of little teeny tiny fat droplets. What that does is it increases the surface tension, which means anything that is working on those enzymes and other stuff like that has more available to it to work on. And a lot of this bile products are going to be recycled. And so, in simple sense, it turns big fat globs into little fat globs. This is a form of mechanical digestion. We are not breaking bonds on this one. We are just messing with the interaction of chemicals with one another. But these are non-bonds. These are not bonds. So this is not chemical digestion. Your gallbladder can be removed. The liver still does make some bile. You just don't have as much. So the ability to eat really fatty foods is going to be decreased with the loss of the gallbladder, but sometimes you can get gallstones, which will lead to a buildup of precipitate in there. And when it contracts, it hurts. Because bile is partly the bilirubin salts from the breakdown of hemoglobin that we talked about before, it does give it that kind of greenish color to it, with when bacteria mess with it, that brownish color that we know feces look like. Lack of bile in the feces will lead to a gray or whitish feces. And again, it's that bilirubin byproduct. Like I said, sometimes you can get gallstones, and like I said, it can be quite painful. The other organ to talk about here is the pancreas. It is kind of shaped like a hand here. You got a wider end and the tail going out. We know that it has endocrine function. We've talked about that this semester already. We're more now with digestion worried about the exocrine function, which is these acinar cells of this. We're going to see there's a main pancreatic duct that travels down here to that hepatopancreatic ampulla, and there's also an accessory duct that goes out to another papilla. So you can see that right here. You have the minor and major duodenal papillas. You can see the pancreas has a main duct going from all these acinar cells to these main openings to send the pancreatic secretions out into the duodenum. What are these making? Well, these acinar cells are making a couple different things. Again, simple cuboidal cells. Main thing they do is they produce a lot of the enzymes that are going to be important for breaking down these materials. So we have a lot of these hydrolytic enzymes. So there is a pancreatic amylase, there's a pancreatic lipase, as well as a couple of nucleases and a protease that is made by the pancreas. These can be in one of these secretions that are going to do that. Some of the duct cells are going to make and secrete what is called a bicarb-rich solution, an alkaline solution. Again, the kind coming from the stomach is going to be highly filled with hydrochloric acid. That is a very low pH. The small intestine, much like the esophagus, does not have the protective mechanisms in place. So what we do is we release substances that are going to neutralize that acidic pH. And the main one here is this pancreatic bicarb-rich solution. Again, it's these acinar and duct cells that are making that. Very alkaline, mostly water. But again, pancreatic amylases to digest starch, pancreatic lipases to digest fats. Things like trypsinogen and chymotrypsinogen, which are proteases, inactive forms of that, as well as we're going to see a couple different nucleases for digestion of those nucleic acids. So we have something for pretty much all those macromolecules that we could have ingested. Again, the CCK, much like it acted on the gallbladder, acts on the pancreas as well. So it's going to stimulate the pancreas to release the pancreatic juice, and causes that to get dumped into the duodenum as well. So again, CCK stimulates the gallbladder to dump its content, stimulates the pancreas to insert its contents, as well as tells the stomach to kind of slow its motility and release of gastric secretions. So now the small intestine can start working on that, and we've added pretty much everything in that we need to start breaking this stuff down. Another hormone that is released from the intestines is secretin. Secretin acts on those ductal cells of the pancreas to release the alkaline solution, that bicarb-rich solution, which helps to neutralize that acidic chyme and makes it so that the acidity from the stomach does not cause damage to the small intestine. So again, pancreatic juice, mainly water, salt, sodium bicarb, as well as enzymes. Again, going to be those hormones CCK and secretin that are going to trigger the release of this, and again, influenced by that parasympathetic nervous system. And again, you can see stuff we've already talked about here, but on a nice little figure here showing the acidic chyme entering. We know those intestinal endocrine cells release secretin. Secretin travels to the pancreas, tells the pancreas to release its bicarb-rich secretion, which is going to neutralize that acidic chyme coming from the stomach. So what we'll look at the next time is kind of finish off some of this. Be looking at the end of the digestive tract, so the colon. And then the last thing to talk about is really breakdown and absorption of some of these contents. So I will see you next time.